Hello everyone, myself Mohammad Muzaffar, Assistant Professor, School of Pharmacy, Nella Narasimha Reddy Educational Society. I'll be dealing with a microbiology subject which is in the first semester of second years, which is pharmaceutical microbiology. As per the PCI syllabus given to you, the whole subject is divided into 5 units in which you'll be learning introduction to microbiology, what are various microorganisms, how can they be isolated, cultured, identified, and in brief we'll be studying what are bacteria, what are viruses, what are fungi. We'll also come across certain rDNA technologies and how vaccines are prepared, what are various vaccines produced, and what are the identification techniques for vitamins and antibiotics. First unit deals with the introduction to microbiology and various microorganisms and their structure, isolation, identification, and uses. So, so I'll be making a series of lectures on various topics separately based on their units. Today's first video is about the introduction to microbiology. Before I get into the topic, let us see what are the objectives of today's class. As you know, microbiology is a branch of science, isn't it? Biology means the study of living organisms, and microbiology means the study of living organisms which are in micrometer of the size, which are tiny sized organisms, isn't it? So, as every branch of science has its definition, similarly, microbiology has a specific definition. So, first we'll be dealing with the definition of microbiology. Next will be the characteristics. As you know, every organism has certain characteristics. Suppose let us take an example of a mobile itself. If we have to identify mobile from a given gadgets, electronic gadgets, you'll first look at the appearance of those electronic gadgets. For example, for the mobile, it will be a front and back camera, smaller size when compared to other gadgets such as laptop and iPads, etc. So based on that, our mobile will have a smaller size, front and back camera, a slot for the SIM, a USB cable attachment, the other side one, and a port for headphones, and so on. These will be the characteristics of a mobile in order to identify the mobile in a given gadget. Similarly, microorganisms are different from human organisms or different from animals, birds, insects, right? So there are certain characteristics. So we'll be dealing with the characteristics of the microorganisms. Next one, as I have said you the example of mobiles, similarly there are various kinds of brands in mobiles. It could be Oppo, Vivo Plus, it could be Samsung, or it could be Lenovo. There are various brands of mobiles, isn't it? Similarly, there are different classes of microorganisms such as bacteria, fungi, and these are based on their taxonomy, and there is a classification based on their effects on the host cell. So we'll be dealing with the classification, and then everything has a history. It could be either a mobiles have also have history. It could be human have history that is from the early man, isn't it? Similarly, viruses or bacteria or any other microorganism, they have certain history. Who has discovered it and how did they identify it, and what are their theories? All those will be seeing in the history, not the complete history, just the important scientists who has contributed their work in microbiology. And then we'll go on with the applications of microbiology, various applications in various fields of science. Okay, so beginning with the definition, just focus on the highlighted words. I have said microbiology, isn't it? So biology is a branch of science, right? So it is a branch of science. And what do we study in microbiology? It will be a study of microorganisms. So it is a study of microorganisms. Then microorganisms are very small, so they are not visible to naked eye. So the next point is not visible to naked eye. And in order to see those microorganisms, we need something to magnify their structures, isn't it? Something to magnify their body, and that will be a use of a microscope. So let us have a definition now. It is a branch of science that deals with the study of microorganisms, isn't it? It is a branch of science that deals with the study of microorganisms which cannot be seen by naked eye, right? It cannot be seen by naked eye, but can be visualized having a microscope, or needs a microscope to visualize them. Let us repeat the definition again. It is a branch of science that deals with the micro study of microorganisms which cannot be seen by naked eye and needs a microscope to see or visualize it. So this will be the definition of microbiology. Moving on to the characteristics, we have seen the definition that it is a branch of science that deals with the study of microorganisms, and I think you are aware what micro stands. Milli means 10 power minus 3, whereas micro means 10 power minus 6. Therefore the microorganism size will range starting from 10 power minus 6, and it can go up less than 10 power minus 6, even 10 power minus 7, 10 power minus 8. It depends. So there is no chance of a microorganism to have a size of millimeter, right? Mm. It is impossible for a microorganism. If anything is in millimeter, it is called as insect, not a microorganism. Okay. And first is size, and from the definition we have seen it cannot be seen by naked eye. So the next characteristic is not visible to naked eye. And I've also mentioned we need a microscope to visualize it, so we need a microscope. This will be another characteristic. Every organism, let it be human or animal or plant, all are multicellular, isn't it? We have a number of cells which continuously gets regenerated and continuously gets degraded or destroyed. So there is a continuous growth of cells, and there are various types of cells in our body, similarly in plants. While in the organism, it is not must to have many cells or different types of cells. It could be have a single cell type, or multiple cell type. Therefore it can be of either multicellular or unicellular. Unicellular examples can be amoeba, paramecium, whereas multicellular can be the bacterial cells, and various saprophytic things like fungi, spirophytes and all. So we'll see in detail what everything are. There are certain bacteria, or there are certain microorganisms which can survive in the absence of oxygen, and they survive in the presence of oxygen. All humans are meant to survive in presence of oxygen. If there is no oxygen, humans can't survive, neither animals nor plants. But it is not so with the microorganisms. They can survive either in presence of oxygen or the absence of oxygen, and therefore based on that they are called either aerobic respiration, sorry, they are called either aerobic bacteria or anaerobic bacteria. Aerobic microorganism or anaerobic microorganism. Now, let us talk something about distribution. It is not necessary that a bacteria to be in particular place. It can be anywhere. It could be in our saliva, or it could be on the benches that we sit. It could be on the things that we use, such as mobiles. Even mobiles can have a bacteria because we place it somewhere. Even on the in the soil that we sit on, or in the soil there where we stand, or in the soil where we play, it can have a bacteria. And it can also be present in water that we drink. It can also be present in the food, and therefore it causes food poisoning. So there's no specific place for the bacteria. It can stay wherever it wants. Therefore, the microorganisms are present everywhere. It could be in air, water, soil, surface of the human skin, or on the plants, or on the animals, or it could be even on the places, various places like benches, chairs and all. Okay, these were the characteristics of microorganisms. Let us deal with the classification. As I have said, every microorganism has specific function. Let it be which is eco-friendly, or let it be one which causes a disease. If the microorganisms are eco-friendly, that which means they doesn't cause any harm either to the host cell or to the surroundings it, they are called non-pathogenic. Non-pathogenic means those organisms which doesn't cause any harm to the host cell. On the other hand, pathogenic microorganisms. Pathogenic microorganisms are those which cause harm to the host cell. For example, let us consider an example of Plasmodium vivax. Plasmodium vivax is pathogenic. When it enters the human beings, it causes malaria, isn't it? Similarly, Mycobacterium tuberculosis, which causes tuberculosis, Vibrio cholera, which causes cholera, Salmonella typhi, which causes typhoid. All these are the pathogenic microorganisms, right? Whereas non-pathogenic are those which doesn't cause any harm. For example, we can take curd. Curd has many lactobacillus species, and this curd is very much helpful for the digestion of food, isn't it? And there are many E. coli, Escherichia coli in our intestine, which are helpful in digestion of green materials, cellulose kind of things. Not all Escherichia coli are good for human beings. There are certain strains which are good for human beings, and there are certain which cause cancer in human beings. So let us see the points, or let us see what are the advantages or disadvantages of them respectively. In case of non-pathogenic, as we have said, harmless, they doesn't cause any harm to the host cell, so they are harmless, can be beneficial, that means eco-friendly, and many live on the surface of the skin, okay. And saprophyte live on a dead matter. Saprophytes that live on dead matter. These microorganisms are the scavengers of the dead organisms that are on the earth. If those organisms aren't there, it is impossible for a dead matter to be decayed. And 70% of the bacteria which are present in the world are non-pathogenic, whereas only 30% are pathogenic and cause various diseases. Pathogenic, as I have said, cause disease and cause infection. They have different shape, distinct shapes, could be rod shape or spheroidal shape, thread like. Well, that will deal the morphology of bacteria and viruses separately in different video. And some produce toxins. These toxins are responsible for various diseases in human beings and also in the animals. For example, Bacillus anthracis. This is an organism which called anthrax in cattle. That is a disease which leads to the death of animal if not treated. And these pathogenic bacteria can be easily spread. The best example can be uh, coronavirus. It is a virus, isn't it? And you have been observing in what speed the cases are increasing, the infection is spreading, isn't it? And another point to remember is they live everywhere, as I have discussed earlier. Let us move on to the classification in other way, that is based on the taxonomy. So based on the taxonomy, microorganisms can be of four types. They are bacteria, and you can see the shape here, cocci and spirochetes. There are different shapes of bacteria that we'll deal in the next video. And fungi, you can see this kind of fungi on the bread, the gray color spores kind of thing, that is called fungi. And protozoa, these are unicellular, amoeba, paramecium. Algae, these are chlamydomonas, spirogyra. And viruses, there are different viruses, we'll deal it with it in later videos. Now let us have a history of microbiology. As I have said, everything has a history. Let it be human beings or animals, they have certain history. We have evolved from early man as per the science, right? As per the Charles Darwin theory of evolution, we have been evolving from early man, and now we are at a stage of human being which is highly evolved organism. Similarly, animals do evolve. Now we'll have a look in the microorganisms which have been evolving and what were the contributions of various scientists around the world in the past. We'll be dealing only with the important scientists. Every scientist is important, but the major contribution who has done, we'll be discussing about them for today. Beginning with Francisco Redi. Francisco Redi is a Italian physician. It means he was a doctor in Italy. Okay, and he, in the beginning, people used to think that organism will come into existence just by itself. just like there is no source for the organism to come. It just come, like it comes just like that. It was believed earlier, but Francisco Redi was the first one to disprove that theory. He was the first one to disprove theory of spontaneous generation of life. So for this, he has performed an experiment in 1668, that is in the 17th century. In his experiment, he has taken a jar and put a meat in that. He has taken three jars. He has opened the mouth and put a meat in that. The other one, he has covered it with a porous cloth, and the third one, he has completely sealed it with the meat inside it. He has been observing for a few days, and there he was able to find that this meat was having larvae grown on it, as well as flies were sitting on it. He understood that these flies were depositing eggs on the meat. As a result, larvae were growing. In this case, the flies were sitting on the cloth and laying their eggs. Those eggs were falling on the meat. As a result, he was able to see only the larvae but not the flies inside the flask or inside the jar. In this case, there was no chance for the fly to get into, nor do it has any chance for the eggs to drop into it. Therefore, this meat was safe. By this experiment, he has proved that the decay of an organism or decay of a nutrient medium is not by itself. It is because of the microorganisms present in the air. He also proved that the growth of microorganism, that means the growth of a new life, the biogenesis, isn't it? The growth of new life is not by itself, but it is because of some source. And what was its source? It was either the flies or it was the air which was carrying eggs of flies. So it was his great contribution in microbiology. And moving on, Anton von Leeuwenhoek, in the same 17th century, he was the first man to discover a microscope. He was actually a textile businessman. He has a business of cloth trading, and he has a very much curiosity towards observing the things which are very small. So what he used to do is he used to take a beaker and fill it with water, and he used to observe through the water so that the things which are on the ground can be magnified and visualized. So based on this observation, he has bought many other glasses and have made a single lens microscope. With that, he was trying to observe many small organisms. He used to observe the quality of the cloth with that microscope. And at the same time, he had made a microscope. You can see the image here. In this image, this is the holder for the glass slide, which has a microorganisms, okay. And he used to observe from this side, and there used to be a source of light from here. You can see in this image there is a light source here, and he used to hold it and observe through the small point where he can see the slide. And based on that, he used to see how the organisms were moving and what were the structure of those organisms. So based on this observation, he first observed bacteria in 1976, okay, using his own design of a microscope, which was this one. And due to his contribution, he was called as the first microbiology in the history of microbiology. And when he first observed the small organisms which were moving from one place to another, he called them as small molecules, small animals which are able to move from one place to another. He has given another name in Dutch, which means animalcules in English. So he has named those tiny animals as animalcules. Next, Louis Pasteur in 19th century, 1882 to 1895 was his time period of his survival. It was his lifespan. He was a French biologist. That means he was very much keen into learning more and more about the living organisms, right? In French, he was the first one to prove or disprove the theory of abiogenesis, and he was the first one to prove biogenesis. Okay, this theory, what it is, will deal now. As per his theory, he always says that life comes to existence when there is a certain As per his theory of biogenesis, he always says that, similar to that of Francisco Redi, that a nutrient medium or any organism can be decayed only when it comes in contact with certain microorganisms which are in the air. To prove his theory, here what he did is, please look at the image here. You can see a flask where he has added a nutrient broth. This is a nutrient medium. In this nutrient broth, he has attached a S-shaped flask or S-shaped connector, which is called as swan neck flask, and he has heated so that he can sterilize it, and then he has boiled it so that he can sterilize the whole nutrient medium. Then he allowed the air to go out so that there won't be any air in that. Definitely there will be some kind of air, but all the uh, steam goes away, and he allowed it to cool. Then he kept it for some time, not for some time, for few days. During this time he was able to observe that in this position, in this place, microorganisms were settling, they were accumulating. What could be the reason? It was because of gravitational pull. Through the air, the microorganisms would get into it. As a result, they would coagulate here or they would accumulate here. They have no further energy or they have no further capability to go against the gravity. Therefore, they were not able to move upwards. Okay, and he was observing that one end of the S-shaped neck flask or S-shaped neck connector has microorganisms. Now the same flask, he has put it in a tilted position or inclined position and allowed the nutrient medium to come here, and after some time he was able to see that the whole solution was turbid, whole nutrient medium was turbid, which means the entire nutrient medium was contaminated by microorganisms. By this he said that nutrient medium will be contaminated only when it is exposed to air that has microorganism with it, and he also proved by this that air also contains microorganisms in them. This was his theory of biogenesis, which is a disproving theory of abiogenesis. He has also contributed many things. One among them is pasteurization. As you know, pasteurization of heating of milk to remove microorganisms. Not only milk, it could be heating of any food items to remove microorganisms. He also improvised the method of fermentation. In the wine industry during his period, uh, during production of alcohol, people were able to see that alcohol was getting spoiled very easily. What could be the reason? Upon doing research, many of them were not able to find out. When Louis Pasteur took took the project and tried to find the reason behind it, he was able to find two things. First thing was a yeast present in the alcohol as well as rot-shaped bacteria. He then confirmed that the presence of rot-shaped bacteria is the reason for the spoilage of alcohol, whereas yeast produces alcohol from various sources. On the other hand, it was a rot-shaped bacteria that was spoiling the alcohol. Okay, he improvised the method of fermentation, and he also introduced pasteurization, and he was the first one to introduce vaccines, especially for anthrax. Okay, and also for cholera. He was the first one to discover vaccines. Moving on further, Robert Koch in 19th century, from 1843 to 1910, it was his lifespan. He was a German physician, and he has developed many techniques to identify the microorganisms, that is by staining techniques. So he was the first one to develop a relationship between a disease and disease causing organism. For example, malaria is a disease. How is it caused? It is caused by plasmodium vivax. How can we prove that? So he has done an experiment where he can prove what was the reason, or what how the disease is responsible, how the diseased organism is responsible for the disease. In his experiment, he has first taken a cattle. Okay, this is, you can see the image here. He first took the cattle and then took a which is infected with anthrax. Anthrax is caused by Bacillus anthracis microorganism. So he has taken the blood sample of the cattle, and he was able to observe microorganisms in them, that is Bacillus anthracis. He isolated it, cultured it, and that cultured microorganism or Bacillus anthracis he injected into the rat. After some times, he was observing the same symptoms with that of the cattle in the mice, and therefore he has isolated blood and then observed under microscope for the bacteria present, and he was able to find the bacteria. So that bacteria he has taken back and isolated it and compared with that of the bacteria which isolated from the cattle, and he was able to see the same kind of bacteria, that was Bacillus anthracis. By this experiment he has postulated four things. We'll see now. First postulate, you can see here, microorganism must be found in all cases of the disease. Doesn't matter which disease it is, microorganism will always be present, whether it could be a bacterial infection or viral infection, fungal infection, anything it could be, and that will be because of microorganism. So every disease has microorganism within the patient or person suffering from that disease. Next postulate that he made was, it must be isolated from host and can be grown in pure culture. From his experiment, he has first taken the microorganism from the cattle, isn't it? From the cattle, he has taken the blood sample and observed under microscope for bacteria, that is Bacillus anthracis, and then isolated and cultured it. That means he was able to isolate isolate the microorganism and culture in a pure culture, and grow in a pure culture. That means he was able to prepare, right? He was able to prepare as well as maintain it in a pure culture. So second postulate is, it must be isolated from host and can be grown in pure culture. Third postulate is, it must produce the original effect or original disease when injected into the host. From the experiment you have seen, the microorganism were first isolated from the cattle, and then that was cultured. Then the cultured microorganism were injected into the mice, and that mice also have shown the same disease, that is anthrax. So cattle and the mice both have shown the same disease, that is anthrax, and the positive organism was Bacillus anthracis. That means in the cattle, if it was one generation, upon culturing he has produced many generations of the same bacteria. So first generation caused was Bacillus anthracis and caused the anthrax. The second generation, or let it be third generation, that is also caused anthrax in mice. That means doesn't matter which generation it is, doesn't matter which organism it is, it causes same kind of infection in host cells. So you can see this, it must produce the original disease even several generations when injected into the host. Now the last, same organism must be recovered from the experimentally infected host. What does it mean? As per the experiment, he first isolated a bacteria from the cattle, right? He cultured it and given it to the mice. It is not a condition where from the cattle he got a different bacteria and from the mice it was a different bacteria. It was not the case. It was the same bacteria that he isolated from the cattle as well as from the mice. That means there is no difference. So from the experimentally, from the mice is an experimental condition, right? Experimental method. He injected into the mice, that was the experiment. So from the experimental object, from the experimental animal, he was able to isolate the bacteria and culture it. So the last point means, same organism can be recovered from the experimentally infected host, that was from the mice. So based on this postulates and his contributions, he was also given a title as father of bacteriology. So now moving on to the applications. These were the important scientists that has played a major role in the field of microbiology. There are various other scientists also, but just for a brief introduction thing, I have considered only four scientists which has brought revolution in the field of microbiology. Moving on to the applications. First application of microbiology or pharmaceutical microbiology can be genetic engineering. In genetic engineering, what they do is, by the term you can understand genetic engineering, that means some engineering stuff will be done in the genes of an organism. So they'll take an organism like microorganism and take their nucleic acid, could be DNA for bacteria or RNA for viruses. They take the gene, alter the gene, and see for the possible results that they would get. They could even make a vaccines out of it, or they can make a bacteria such that it produces more of the antibiotics. For example, penicillin. Penicillium notatum gives penicillin as a metabolite, which is active pharmaceutical ingredient and cause and helps in and acts as antibacterial, isn't it? So they can alter the gene of that penicillin notatum in order to produce more of the metabolite, that is more of the penicillin. Similarly, it is also implied or applied in medicine. How can it be used? Just I have discussed the example of penicillin. Similar to penicillin, cephalosporins can also be generated or produced from microorganisms, and also streptomycin or cyclosporins. All these are the antibiotics that are produced from microorganisms, and these antibiotics are most potent one. And in agriculture, especially the bacteria used for nitrogen fixation, you can take an example of leguminous plants. The root nodules has a bacteria called as rhizome. This rhizome helps in fixing the nitrogen within the soil from the atmosphere. There are various other microorganisms also that helps in that is implied in agriculture. Now in industry, various microorganisms are used to produce antibiotics, vitamins, and vaccines, right? And the most importantly, yeast is used to produce alcohol, as we have discussed in Louis Pasteur's case. Yeast is used for the production of alcohol as well as in bakery to produce to prepare breads and dairy products. And last, in research, we might not know how the research actually goes on. So what researchers do is they first look at the nucleic material of the microorganism and study their structure, the protein part that they have, capsid and all, the bacterial cell wall. They study everything and find the reasons for the toxins that are being released from those organisms that cause disease in the human body, and they try to alter those genes, and so that they doesn't produce any uh, or they doesn't produce any metabolite, or they reduce the release of toxins from the microorganism in order to reduce its action on the host cell, as well as it can be used to produce new vaccines and new antibiotics, or increase the production of antibiotics from the microorganisms. All these are the applications. First is genetic engineering by RDNA technology, in medicine preparation of antibiotics, in agriculture nitrogen fixing and many other uses, and industry in preparation of alcohol and dairy products, as well as in bakery, and in research for altering the genes and production of new vaccines and all. So this was the introduction to microbiology. From this class, you might have learned the definition of microbiology, right? And we have seen the characteristics of all the microorganisms. Then we have classified microorganisms based on their nature against the host cell or host organism, and we have also classified based on the taxonomy. And after that, we have seen history, a small part, a brief one, and then we have seen the applications. So in the next videos, we'll be seeing in brief the structure of bacteria, the ultrastructure of bacteria, I mean to say, and how can they be isolated, how they can be identified, and what are the uses, and what are their disadvantages. I mean, what are the organisms which are beneficial and what are the organisms that are harmful, and how uh, people are able to, or how the researchers across the globe are able to isolate bacteria. All these we will be dealing in the next videos, one after the other. So thank you for watching the video.